It's really a pleasure to be here, and thank you so much for having me to talk about Adaptive Biotechnologies ImmuneCODE database and the related immunoSEQ TMAP COVID functionality. Adaptive is a commercial immunosequencing company. We've been providing this service since 2009, and this has developed into a couple different focuses within the company, but today we'll be focusing on the immunoSEQ and our TMAP COVID. If you're not familiar with our offerings, we do offer a variety of assays for different loci in both humans and mice. And if you'd like to learn more, you can contact us at research_sales@adaptivebiotech.com. But what we want to talk about today is our ImmuneCODE project to map responses to COVID-19. And the data within this ImmuneCODE database is both sequence level information and repertoire level information. The sequence level information is over 135,000 antigen specific T-cell receptors that respond to SARS-CoV-2. This was done with our MIRA platform, which is a multiplex system for identifying antigen specificity. The repertoire level information has come from our own internally sponsored study called ImmuneRACE, as well as partnerships with institutions around the world to sequence and make available the data for individuals exposed to, actively infected with, or recovered from COVID-19. And all of this has come together in that ImmuneCODE database, but also is available for incorporation into your own work in our immunoSEQ analyzer through our immunoSEQ TMAP COVID. With this, you have full access to all of those SARS-CoV-2 specific antigen receptors for the TCRs. You're also able to directly incorporate this data into your own work generated through Adaptive. It can also be exported and worked with used in a variety of the tools we've heard about today. And hopefully will really allow researchers to dive into patient population and cohort level information. All of this data is publicly available through our Immune Access platform. You will have to create a account within our immunoSEQ analyzer, but again, that is completely free, and then you will have access to everything shown here today. In order to access this data, you can go to Immune Access if you already have an account and are logged in, or you can go directly to clients.adaptivebiotech.com/pub/COVID-2020. However you access this data, you will be able to open the data in the analyzer by using that Open in Analysis link highlighted in the red box. This will open all of the repertoire information and allow you to work with it directly in our software. If you want to download any of the information and use it offline or in another program, scrolling down to the bottom of the project, in the lower right-hand corner, there will be a list of supporting documents and links. The top one listed there, which is the MIRA release, is going to have all of the epitope information, as well as that over 135,000 sequences that you can download and work with. And I'll show you some examples of how those files look in just a moment. You'll also be able to download all the metadata that's associated with these different cohorts and samples, so you can see what information is available and maybe which cohorts or samples you'd like to focus on with your research. You're also able to download all of the repertoire information all at once. This is going to be a large file format, even compressed, it's close to 25 gigs of data. There are over 1,400 repertoire samples in there from a variety of individuals across the world. An example of how some of this data is going to be delivered to you. Within the MIRA files, you will have a file about peptide hits. All of these are going to be available as CSVs that can be opened in Excel or a variety of other programs. For the peptide hits, there's information about the reading frame or protein that the peptide comes from, as well as the actual epitope sequence, its location in the genome, and then that hits column all the way on the right is how many TCR receptors actually responded to those peptides. You will also have access to all of the immunosequencing information for the specific T-cell sequences. In this detailed file, there will be information about the T-cell bioidentity, which is the amino acid sequence of the CDR3, as well as the flanking V and J genes. You'll also have the nucleotide sequence, as well as what reading frame and what specific epitopes that sequence responded to. If you open the data in the actual analyzer, you're able to see all of those over 1,400 samples, as well as their associated metadata, all the sequencing information, and you'll have full access to all of our features, functions, and tools to analyze this data. One of the new tools we're really excited to have is the TMAP COVID report. This allows you to blast any of these samples or any of your own samples against that large database of known antigen-specific T-cell receptors, and it will annotate those sequences in your sample that are SARS-CoV-2 specific. As an example, here is one of the TMAP COVID reports. This will list every clone within your sample and annotate each clone. So it's going to give that bioidentity and the nucleic acid rearrangement for your clone, as well as the template count, so how many cells express that receptor in your sample, the productive frequency, all of the information for all of your sequences. And then it's going to go through and actually annotate each of those clones. So it'll let you know which open reading frame or protein it was responding to, as well as the specific epitopes on which it hits. If you want to learn more about this or stay up to date when we have new data releases, you can go to immunerace.adaptivebiotech.com. There will be an introduction to ImmuneCODE, links to find the various publications about this, where you can learn more about the database and what all is entailed in it. There's also up-to-date information about our ImmuneRace study, how many people have been enrolled, the different repertoires, and some of the metadata, as well as blog posts and data release updates. So with that, I think we might have time for questions, but if you have any questions or would like to find out more, please don't hesitate to reach out to us at research_sales@adaptivebiotech.com. So thank you again, everyone, for listening, and thank you, AIR, so much for having us today.
Thanks so much for the very nice talk. So maybe we have time for 1 or 2 questions. I will wait a bit so that the questions can come in. People are a bit shy today.
Yeah.
So maybe I can ask the first question. So maybe I've missed this, but for the epitope-specific data, do you provide, is it only TCR beta or also the alpha chain that is provided? So is it?
So currently it's only TCR beta information.
I see.
We do, however, have the TCR beta information broke down, into the context of HLA class 1 or class 2.
So we have a question from Frederick Madsen, who asks, can you describe the machine learning approaches that you have applied so far to these data?
Yes. So we haven't, we actually have a whole publication about some of the different conclusions that we've drawn from this data, but really where the machine or computational approach has come in is identifying those MIRA clones. So MIRA assay is highly multiplex, and then we computationally are able to link epitopes to specific T cells. Then as far as machine learning, we are actually about to fully launch a diagnostic called T-Detect that will predict, based on those T cell repertoires, past exposure to COVID.
Very interesting.
And I'm sure we'll have much more information about that once it is fully launched.
Very nice. And then one last question. Are any public CDR3s among the anti-COVID CDR3s?
That's one thing that a lot of people are looking at. I don't know the exact number of public clones, but there are clones that you can find between different individuals. How many or their frequency is one of the reasons that we're making all this data publicly available so people can investigate that.
Great. I think we have to move on, but thanks so much for the wonderful talk. And as I said, people can reach out to you also after.